You can drink coffee every single morning, feel completely energized, feel like you are doing something right for yourself, while the most powerful cellular repair window your body runs is quietly slamming shut before you finish your first cup. The damage is not in the coffee. It is in when you drink it and what you put in it. And the two habits responsible have been shaped for most people by 40 years of marketing dressed up as nutrition advice. By the end of this, you will have a three-part framework I call the three-window reset, something that costs nothing, requires no new products, and works with the biology your body is already running every single morning. If you make it to the protocol section, you will understand exactly why this works and why you have never heard it explained this way before. Let me start at zero. The moment your eyes open, between midnight and 6 in the morning, your body has been running what I think of as the overnight maintenance shift. Not just sleep, a coordinated biological overhaul that most people never think about because it happens invisibly, quietly, and completely outside their control. Your growth hormone levels peak during the first cycle of deep sleep. Your liver, which spent the evening processing everything from dinner, shifts mode somewhere around 2 in the morning and begins releasing stored glucose at a controlled rate to keep your brain fueled without any incoming food. Your immune system runs a surveillance sweep through tissue that is harder to access during waking hours when blood flow is distributed across active muscles. And in every cell in your body that contains a nucleus, which is essentially every cell except red blood cells, a process called autophagy has been running since approximately 10 to 12 hours after your last meal. Autophagy. The word comes from the Greek for self-eating, which sounds alarming and is actually the single most important cellular maintenance mechanism your biology runs. Here is what it actually is. Inside each of your cells, there are specialized cleanup units. Picture a mobile garbage disposal operating inside a tiny room. These units circulate through the interior of the cell, identify damaged proteins, broken or dysfunctional mitochondria, and accumulated cellular debris, and dismantle them for parts. Those parts get recycled into new building blocks. This is not a health metaphor. This is a specific, Nobel Prize-winning biological mechanism. In 2016, the Nobel Prize in Physiology or Medicine was awarded to Japanese cell biologist Yoshinori Osumi for mapping exactly how autophagy works. The research established that when autophagy slows or stops, cellular debris accumulates. And that accumulation, that uncollected garbage inside your cells, is directly linked to accelerated aging, chronic low-grade inflammation, and the cellular dysfunction that shows up years later as metabolic disease, cognitive decline, and muscle loss. After 55, autophagy runs slower as a baseline. Slower startup, lower peak intensity, shorter active windows before the cellular maintenance signal fades. This is not a catastrophe. It is a biological reality that changes how sensitive your morning choices need to be. And the thing most commonly disrupting that window for people in this age group is something they have been told is healthy or have simply never questioned. Autophagy thrives in a fasted state. When your body is not processing incoming food, it redirects cellular energy toward maintenance rather than construction. The presence of nutrients, particularly carbohydrates and fats, triggers insulin release, and insulin is the master off switch for autophagy, not a dimmer, an off switch. The moment insulin rises, the body redirects resources toward fuel processing and storage. The cleanup crew clocks out. Whatever cellular debris was waiting in the queue stays in the cell. The invisible villain in most people's mornings is the calorie signal, and it arrives, for tens of millions of people, inside something they consider a near sacred morning ritual: their coffee. Specifically, what goes into their coffee: cream, oat milk, almond milk, a flavored pod, a splash of half and half. The product marketed as fasting compatible because someone on the internet said fat does not spike insulin. Let me tell you what the endocrinology actually says. Any caloric intake, including the 15 to 50 calories in a tablespoon of cream or milk, triggers an insulin response in people with normal pancreatic function. The response is dose-dependent, but it is not zero. And for people over 55, whose baseline insulin sensitivity is measurably lower than it was at 35, the threshold for disruption is actually lower, not higher. The system has become more reactive, not more forgiving. More reactive, meaning a smaller caloric signal produces a proportionally larger metabolic interruption. The margin you had at 40 to make small exceptions to the fasting window is narrower at 55, and it narrows further each decade. So the calorie villain is clear. But the second villain, the one almost nobody is talking about, is timing. Even black coffee, with 0 calories, creates a specific problem when consumed immediately after waking. And understanding this problem requires understanding something that almost never appears in mainstream wellness content: the cortisol awakening response. Cortisol is a hormone most people associate with chronic stress, and that association has turned it into a dietary villain in popular health media. Cortisol is bad. Cortisol causes belly fat. Cortisol kills your gains. But cortisol has a critical scheduled morning function that has nothing to do with stress responses. Every morning, between roughly 15 and 45 minutes after waking, the adrenal glands produce a sharp and deliberate spike in cortisol, somewhere between 50 and 100% above baseline. This spike is a scheduled metabolic event. It activates the immune system for the day. It raises blood pressure slightly to support alertness. It elevates blood glucose just enough to fuel the brain without food. It primes the metabolic machinery. Researchers at the University of Trier in Germany, who have studied this phenomenon for decades, found that the amplitude of this spike predicts cognitive performance, immune competence, and metabolic activation throughout the rest of the day. A strong cortisol awakening response is associated with sharper working memory, faster reaction time, and better blood glucose regulation. A blunted one is associated with fatigue, brain fog, and dysregulated appetite. Here is what caffeine does to that spike. Caffeine stimulates the adrenal glands. When you drink coffee within the first 30 minutes of waking, your body receives an artificial adrenal signal at precisely the moment it was preparing to generate a natural one. The two signals do not add, they compete. The body, detecting that the adrenal system is already stimulated from caffeine, downregulates its own natural output. The cortisol awakening response is blunted, flattened. You feel alert. The caffeine is doing its job, but you have just traded your most powerful natural metabolic activator for a weaker, externally generated substitute. And you have done it every single morning. Over time, the cortisol awakening response loses amplitude from disuse, the same way any biological system that is repeatedly bypassed becomes less robust. A review published in the journal Frontiers in Endocrinology in 2023 examined habitual early morning caffeine consumption and found a consistent association with lower cortisol awakening response amplitude. Lower amplitude means less metabolic activation, less immune readiness, and, critically for people over 55, a faster collapse back into fatigue once the caffeine clears. That mid-morning energy crash that most people manage by pouring a second cup is not caffeine tolerance. It is the natural cortisol window that was never allowed to complete its cycle. The second cup compensates for something the first cup prevented from happening in the first place. Think of the cortisol awakening response as a diesel engine warming up before a long drive. It takes 15 to 30 minutes to reach full operating temperature. Once it does, it powers your metabolism, your immune surveillance, and your cognitive function with remarkable efficiency for hours. Drinking coffee at minute 5 after waking is like injecting fuel into an engine that is still cold. The engine moves, but you are burning fuel without getting the full output. And every morning you do this, the engine's ability to warm up independently becomes slightly less reliable. After 55, this matters more because the cortisol awakening response amplitude is already lower than it was at 40. Research published in the journal Psychoneuroendocrinology documented measurable declines in cortisol awakening response with age, with the steepest drops occurring between 50 and 70. The buffer you had at 35, where you could short-circuit the natural cortisol spike with immediate caffeine and still function reasonably well, is gone. The biology is no longer forgiving enough to absorb that inefficiency without cost. So two processes are being suppressed simultaneously in a typical morning for someone over 55. The caloric addition to coffee ends the autophagy window, and the immediate timing of even black coffee blunts the cortisol window. Together, they eliminate two of the most powerful biological repair mechanisms the body runs, both of which were working hard throughout the night and needed only a brief window of morning restraint to complete their work. Now, let me tell you what the adenosine science adds to this picture, because this is the mechanism that makes delayed coffee not just better for your hormones, but also biochemically more effective as a cognitive tool. Adenosine is a molecule that accumulates in the brain as a byproduct of neural activity. Every time a neuron fires, adenosine is produced and released into the surrounding space. Think of it as metabolic exhaust from brain work. As adenosine levels rise throughout the day, it docks into receptors on brain cells and produces a slowing signal, the progressive feeling of mental fatigue that, left unchecked, leads to sleep. Sleep clears adenosine. That is its primary restorative function, which means when you wake up, adenosine levels are at their daily minimum. Caffeine works by fitting into adenosine receptors. It is structurally similar enough to adenosine to occupy the docking station and blocking the slowing signal. But here is the mechanism most people have never considered. The effectiveness of caffeine's blocking depends on how much adenosine it is displacing. At minute 5 after waking, adenosine levels are still low from sleep clearance. Caffeine arrives at mostly empty docking stations. The blocking effect is weak. The alertness response is shallow. By minute 90, you have been awake long enough for adenosine to accumulate meaningfully. More docking stations are occupied. Caffeine arriving now displaces a much larger quantity of adenosine, producing a stronger, longer, and more even alertness response. The same amount of caffeine consumed 90 minutes after waking is pharmacologically more powerful than the same amount consumed 5 minutes after waking. Not because the caffeine is different, because the receptor environment it arrives in is different. A neuroscientist at Stanford University, whose research on sleep architecture and circadian timing became widely read in the early 2020s, began recommending exactly this delay based on the intersection of cortisol and adenosine dynamics. His specific recommendation: wait 90 to 120 minutes after waking before your first caffeine, and the performance benefit of a single cup will consistently exceed what most people extract from 3 cups consumed across an early morning window beginning at minute 0. Now let me take a step back and tell you something about breakfast that I find genuinely funny in a depressing kind of way. We have been told for most of our adult lives that breakfast is the most important meal of the day. This phrase did not emerge from clinical nutrition research. It was not published in a peer-reviewed journal. It was the output of a marketing campaign. In the early 1900s, John Harvey Kellogg, the cereal manufacturer, not a physician acting independently, built a commercial empire on the concept that Americans needed to eat immediately upon waking. That morning appetite was a health signal, and that grain-based cereal products were the ideal delivery system for that meal. The campaign was so successful, it became embedded in dietary guidelines, school nutrition programs, and the popular imagination as medical consensus. The actual clinical evidence tells a different story. A 2019 meta-analysis published in the British Medical Journal, covering 13 randomized controlled trials, found no evidence that eating breakfast improves weight management or metabolic health. Participants who skipped breakfast did not overcompensate by eating more later in the day. Contrary to the conventional claim, their metabolic markers did not worsen. In several trials, morning fasters showed improved insulin sensitivity over the course of the study. The most important meal claim was not based on evidence. It was based on industry lobbying that ran long enough to become dietary orthodoxy. The researchers who published that meta-analysis were not fringe voices. They were at Monash University in Australia, one of the most respected clinical research institutions in the Asia-Pacific region. What actually happens when you skip breakfast and extend your overnight fast toward noon, with black coffee as your only intake during that window, is a two-phase biological sequence that the early morning eating habit systematically prevents. Phase 1 runs from waking through approximately the 90-minute mark. The cortisol awakening response completes its full cycle. Your natural metabolic activation runs without competition. Then the black coffee arrives, riding the descending edge of the cortisol wave, with adenosine levels high enough to make the caffeine's blocking effect maximally powerful. You are more alert, for longer, on less caffeine. Your adrenal system has done its work without interference. The engine warmed up before you injected the fuel. Phase 2 runs from roughly 2 hours after waking through noon. This is what I call the autophagy window, and it is where the cellular maintenance becomes serious. Meaningful autophagy induction generally begins around 12 hours of fasting in healthy adults, and it intensifies through hours 14, 16, and beyond. If your last meal was at 7 in the evening and you wake at 6 in the morning, you are already at hour 11 when your eyes open. The 90-minute wait before coffee keeps the window intact. Black coffee, consumed at that point, does not close it, it actively extends it, for a reason that took me a long time to understand, and that most coffee discussions miss completely. In 2014, a team of cell biologists that included the French researcher Guido Kromer, one of the most cited scientists in autophagy research, published a study in the journal Cell Cycle demonstrating that both caffeinated and decaffeinated coffee triggered autophagy in the liver, muscle, and heart tissue of mice within 4 hours of consumption. The trigger was not caffeine, it was the polyphenols in the coffee, specifically a class called chlorogenic acids, that directly activated the autophagy signaling pathway. The caffeine was chemically incidental. The polyphenols were doing the maintenance work. This is the mechanism that almost every morning coffee discussion ignores. You are not delivering just a stimulant. You are delivering a concentrated package of polyphenols, antioxidants produced by the coffee plant, as a natural defense against ultraviolet radiation and insects, directly into a fasting metabolic environment where those compounds find their highest possible cellular target availability. No competing digestion, no active insulin signaling to interfere. The polyphenols interact with liver cells, muscle cells, and gut cells that are in full maintenance mode, and they push the autophagy program further than fasting alone would push it at that same hour. After 55, the specific cellular target that matters most in this context is muscle tissue. And the reason is a phenomenon called anabolic resistance, something your doctor almost certainly has never explained to you because it does not have a pharmaceutical solution, which means it rarely makes it into clinical conversations. Anabolic resistance is the progressive desensitization of muscle cells to the protein signals that trigger repair and growth. At 35, a modest protein signal, 20 grams from a meal, reliably reaches the threshold required to activate muscle protein synthesis, the process by which damaged muscle fibers are rebuilt and reinforced. At 55 and beyond, that same 20-gram signal frequently fails to reach the activation threshold. The muscle cells are harder to reach. The signal needs to be stronger, or the channel through which it travels needs to be clearer. One of the primary reasons the channel becomes blocked is the accumulation of damaged protein aggregates and dysfunctional mitochondria inside the muscle cell itself, exactly the type of cellular debris that autophagy is designed to clear. Research on older adults using time-restricted eating protocols, specifically eating windows that do not open until noon or later, has shown improvements in muscle quality markers without changes in total protein intake. Not muscle mass necessarily, but muscle quality, the efficiency with which existing fibers contract, regenerate, and respond to training stimuli. The interpretation is straightforward. The extended fasting window, by deepening autophagy in muscle tissue, clears enough cellular debris to make the protein signaling pathway more receptive again. The garbage disposal clears the line. The phone starts ringing through. I want to be careful here because this is an area where the research is still developing and individual results genuinely vary. What the studies show is a mechanism, a direction, a biological logic, not a guarantee. And I say this with some personal context. My husband, who spent years overloading himself with work and almost no health maintenance, had a near-fatal aneurysm about 8 years ago. He came out of it with high cholesterol, damaged blood vessels, and a medication list that his doctors framed as permanent. I went into research mode in a way that probably alarmed the people around me, And one of the things that kept coming up in the literature on cardiovascular repair, metabolic reset, and muscle preservation in older adults was the interaction between fasting windows, coffee timing, and cellular autophagy. We restructured his mornings, the timing of his coffee, the extension of the fasting window, the composition of his first meal, always under medical supervision, always tracking. Results varied, and his recovery had many components I cannot disentangle. But his cardiologist, after about 12 months, made a comment without being prompted about markers she had not expected to move. I am not attributing that to coffee. I am attributing it to finally understanding when the biological systems that were trying to repair him actually needed to be active, and stopping the morning habits that were quietly shutting them down. I had read every study, I had taken notes, and I still kept making my morning coffee the same way I always had. Which brings me to something my husband spent months building after I made clear that understanding the science and actually living by it are completely different problems. He had been ahead of me on the practical side for a long time. He had taken everything, the cortisol timing research, the autophagy window data, the protein timing studies, the glucose management literature, and built it into a daily structure, a 30-day companion that he eventually named the Noon Reset Protocol. 2 pages per day, 1 page of targeted science explaining exactly what is happening in your body on that specific day of the protocol, and 1 page of tracker so you can see your own patterns developing. It runs in 4 phases: adaptation, activation, optimization, and acceleration. The first phase is deliberately gentle because the research on why people quit these protocols early is consistent. Day 4 is physiologically specific, and if nobody tells you what day 4 actually feels like and why, it reads as failure when it is actually adaptation. The guide explains that in advance. It includes 25 breakfast meal ideas with exact protein counts, because what you eat at noon after a 16-hour fast is not interchangeable. The anabolic resistance research makes the composition of that first meal genuinely important. You can find it through the link in the description for less than the cost of one restaurant meal. Now let me tell you what black coffee does to your blood glucose, because this mechanism is the one that I think should be in every nutrition conversation for anyone over 50, and it is almost never mentioned. Chlorogenic acids, the same polyphenols that trigger autophagy, also inhibit two enzymes involved in glucose processing. The first is glucose 6-phosphatase, which converts stored liver glycogen into blood glucose. The second is alpha-glucosidase, which breaks complex carbohydrates into simple sugars in the small intestine. When both of these enzymes are partially inhibited, which is precisely what chlorogenic acids do, the rate at which glucose enters your bloodstream after a meal is measurably slowed. The glucose spike is lower. The insulin response is proportionally lower. And because insulin is the primary fat storage hormone, the chemical signal that locks fat cells in storage mode and tells the liver to convert excess energy into triglycerides, a lower insulin response after eating means a longer window of fat oxidation between meals. Research published in the American Journal of Clinical Nutrition found that drinking black coffee with a meal reduced the post-meal blood glucose response significantly compared to drinking water with the same meal. Studies in this area consistently show reductions in the range of 20 to 30%, depending on the population and coffee type. That range is not a rounding error. For someone over 55 whose glucose tolerance has been declining at roughly 1% per year since age 30, as documented in American Diabetes Association data on age-related insulin resistance, a 20 to 30% reduction in post-meal glucose spike is a clinically meaningful intervention that arrives in a cup of black coffee, no prescription required. The mechanism behind the age-related decline is a process called glucotoxicity. When blood glucose consistently runs higher than optimal, glucose molecules attach to proteins throughout the body in a process similar chemically to caramelization. The proteins stiffen, arterial walls become less flexible, kidney filters become less effective, connective tissue loses elasticity, neurons lose their protective myelin coating more rapidly. None of this is dramatic or symptomatic in the short term. It is slow and invisible, and it shows up on your HbA1c, a blood marker, reflecting your average blood glucose over the previous 3 months, long before it produces any feeling you would notice. The chlorogenic acids in black coffee, arriving in a fasting metabolic environment and then carried forward into the breakfast meal, are positioned to buffer this process in two consecutive phases. During the fast, they support autophagy. During the meal, they slow glucose entry. You are getting two separate interventions from the same drink, but only if the timing and the content are right. There is another mechanism I want to bring in here that connects the morning coffee window to long-term brain health, because after 55, cognitive preservation is just as urgent as cardiovascular and metabolic health, and most people are not thinking about them as related systems. Brain-derived neurotrophic factor, BDNF, is a protein that supports the survival and growth of neurons. Think of it as fertilizer for brain cells. It maintains existing neurons, supports the formation of new connections, and plays a protective role against the type of neurological degeneration associated with Alzheimer's and Parkinson's disease. BDNF levels decline with age. They also decline with sedentary behavior, chronic high insulin, and chronic sleep disruption, three conditions disproportionately common in adults over 55. Research from multiple institutions, including Johns Hopkins University, has identified both caffeine and coffee polyphenols as compounds that can upregulate BDNF production. The mechanism involves activation of a pathway called CREB, cyclic AMP response element binding protein, which acts as a master switch for several neuroprotective gene expressions. Caffeine activates CREB through the adenosine receptor pathway. Blocking adenosine increases cyclic AMP, which activates CREB, which triggers BDNF gene expression. The polyphenols activate CREB through a separate antioxidant pathway involving the NRF2 protein. You have two independent mechanisms converging on the same neuroprotective output, and they are most effective in a low-insulin, fasting state metabolic environment. When insulin is chronically elevated, as it is in people who eat through the morning, insulin signaling competes with the CREB pathway and reduces BDNF upregulation. The fasting window removes that competition. This is not small news for anyone over 55. Hippocampal volume, the part of the brain most directly responsible for memory consolidation and spatial navigation, begins declining measurably in the 40s. BDNF is one of the primary mediators of hippocampal neurogenesis, the process by which the hippocampus generates new neurons even in adult brains. A fasted, polyphenol-primed metabolic state occurring during the 90 minutes to noon window creates the hormonal conditions most favorable to BDNF activity. Not guaranteed results, a direction, not a certainty, but a direction that the typical morning eating pattern actively opposes. Now, I want to bring in the circadian angle because there is a layer of this story that connects coffee timing not just to cortisol and adenosine, but to your cellular clock, the biological metronome that governs when your cells perform which functions throughout the 24-hour cycle. Every cell in your body contains a set of clock genes. These genes run on a roughly 24-hour cycle and time-specific cellular functions: when to replicate, when to repair, when to metabolize particular nutrients, when to express certain proteins. The master pacemaker for all these cellular clocks is the suprachiasmatic nucleus, a cluster of neurons in the hypothalamus that receives light input directly from the retina and uses it to synchronize cellular clocks throughout the body. But the cellular clocks can also be influenced by feeding signals. Specifically, when you eat resets what researchers call the peripheral clocks, the cellular timekeepers in your liver, muscle, fat tissue, and gut. When you eat early in the morning, you send a feeding signal that pushes the peripheral clocks earlier. For some people, this is fine, but research from the Salk Institute, specifically from the lab of Satchidananda Panda, whose work on time-restricted eating became one of the most replicated findings in circadian biology, found that aligning your feeding window with the later portion of your waking day tends to produce better metabolic outcomes in adults. The biological reasoning is that human metabolism is historically a morning to afternoon activity, with the digestive and endocrine systems reaching peak efficiency between roughly 10 in the morning and 6 in the evening. Eating before that window, very early morning, places a metabolic demand on systems that are physiologically configured for maintenance, not digestion. Breaking your fast at noon keeps your feeding window within the period of peak metabolic efficiency. The pancreas releases insulin more effectively in the midday period. Gastric motility is faster, gut absorption is more efficient, and the signal sent to the peripheral clocks by a noon breakfast is more aligned with the master pacemaker's own timing signal, light, which by noon has been present for hours and has fully entrained the suprachiasmatic nucleus. You are eating in metabolic daylight, not metabolic dawn. Let me bring this to the composition of the breakfast meal, because the timing and the content interact in a way that makes each one more or less effective depending on the other. When you break a fast of 14 to 16 hours, your gut is in a heightened absorptive state. Digestive enzyme output is primed. The gut microbiome, which has had the entire fasting window to clear fermentation byproducts and reset its composition, is more receptive to incoming nutrients. This heightened absorptive state is brief. Within 30 to 45 minutes of the first meal, it normalizes. So the composition of that first meal after the fast is not interchangeable with what you might eat at 3 in the afternoon. Because of anabolic resistance, adults over 55 need a minimum of approximately 3 to 4 grams of leucine per meal to cross the activation threshold for muscle protein synthesis. Leucine is the amino acid that functions as the master trigger for the mTOR pathway, the cellular signaling cascade that initiates muscle repair and building. 3 to 4 grams of leucine corresponds to roughly 30 to 40 grams of total protein from high-quality animal sources: eggs, chicken breast, salmon, Greek yogurt, cottage cheese. If the breakfast meal is a bowl of fruit, a smoothie built on nut butter, or anything with less than 20 grams of protein, the heightened absorptive state is being spent on nutrition that cannot reach the anabolic threshold. The window is open, the signal is too weak to pass through. This is why the composition of the breakfast meal is as important as its timing. The extended fast creates an unusually receptive cellular environment. The anabolic resistance research tells us exactly what signal is needed to capitalize on it. Anything less than that threshold is not a partial success. It is a missed signal. Now, I want to close with one more mechanism about coffee that is specific to physical performance, because it is the piece that connects the morning window directly to the reason muscle preservation accelerates or decelerates after 55. Caffeine crosses the blood-brain barrier and increases the firing rate of motor neurons, the nerve cells responsible for recruiting muscle fibers during physical effort. In older adults, baseline motor neuron firing efficiency is lower than in younger adults. Fewer fibers are recruited per unit of effort. Peak force output per contraction is lower. Caffeine at peak plasma concentration can increase peak force output by 3 to 8%, according to a 2020 meta-analysis in the Journal of Strength and Conditioning Research. That is a meaningful magnitude when muscle preservation is the goal. Caffeine reaches peak plasma concentration approximately 45 to 60 minutes after ingestion. If you drink your black coffee at 90 minutes after waking and exercise, walking briskly, doing resistance work, or any movement that stresses muscle tissue, between 10 and 11 in the morning, you are training with caffeine at near peak plasma concentration. Your motor neuron recruitment is more efficient. Your perceived effort is lower for the same output, which means you can sustain higher quality movement for longer without feeling like you are pushing harder. For people over 55 working to maintain or rebuild muscle tissue, that late morning training window, fueled by a properly timed cup of black coffee, is one of the most quietly powerful metabolic advantages available, and almost nobody has framed it this way. Now, the 3-window reset. 3 specific components, each one targeting a different biological window, all 3 necessary for the full effect. Window 1, the cortisol window. For the first 90 minutes after waking, no caffeine. Water is not only acceptable but beneficial. Mild dehydration, which is common after 8 hours without fluid, slightly blunts the cortisol awakening response by itself. Movement during this window is actively helpful. Even 5 to 10 minutes of light walking activates the glymphatic system, the brain's drainage network that clears inflammatory proteins accumulated during sleep. But no coffee. Let the cortisol awakening response complete its full cycle. You will know you have waited long enough when you feel genuinely alert without caffeine. For people who have been drinking coffee immediately upon waking for years, the cortisol awakening response has been partly suppressed by disuse. It takes 1 to 2 weeks before it reasserts full amplitude. During that period, the morning window will feel longer and harder than it will eventually feel. That discomfort is not a signal that the protocol is not working. It is the signal that the natural system is remembering how to start. Window 2, the autophagy window. From 90 minutes after waking until your first meal at noon, the only thing entering your system is black coffee or plain water. Not flavored coffee, not butter coffee, not any product marketed as fasting safe that contains caloric fats. Black coffee. The polyphenols continue pushing autophagy deeper. The insulin signal stays at baseline. If hunger is significant during this window, drinking water first will address most of it. Thirst and hunger share overlapping signaling pathways and are frequently confused. 5 minutes of brisk movement also suppresses ghrelin, the primary hunger hormone, for 20 to 30 minutes through a pathway unrelated to food intake. Most hunger felt between 9 and 11 in the morning by people accustomed to eating breakfast is conditioned hunger, a clock response trained by years of eating at that time, rather than physiological hunger. Conditioned hunger passes if you wait it out. Physiological hunger intensifies regardless of distraction. Learning to distinguish the two is a skill that develops within the first 10 days of this protocol. Window 3, the break-fast window. At noon, the first meal should contain a minimum of 30 grams of protein from a high-quality animal source, enough to deliver 3 to 4 grams of leucine, and cross the anabolic resistance threshold. The chlorogenic acids from the morning coffee are still partially active in the gut and liver, buffering the glucose response from incoming carbohydrates. You are breaking the fast at the moment of peak absorptive readiness, with the glucose-blunting polyphenols already in position, and with enough protein to reach the muscle repair threshold that 55-year-old muscle tissue actually requires. The one mistake that invalidates the protocol completely is flavored coffee during window 2. Several popular single-serve coffee pod products, including flavored varieties from major brands, contain 2 to 5 grams of sugar per serving. 2 to 5 grams. That is enough to spike insulin, enough to end the autophagy window, enough to flatten the cortisol to coffee sequencing by introducing a glucose signal that shifts the body back into processing mode. Read the ingredient list. If anything appears beyond coffee, it is not window 2 appropriate. This is not perfectionism. This is the difference between running the protocol and thinking you are running the protocol. One final detail on coffee selection that changes the polyphenol equation more than most people realize. Light roast coffee contains significantly more chlorogenic acids than dark roast. The roasting process degrades these polyphenols. The longer and hotter the roast, the more of them are cooked out. A study published in the journal Food Chemistry found that light roast varieties contained up to 60% more chlorogenic acid content than dark roast varieties of the same bean origin. Stronger flavor in coffee and stronger polyphenol content are inversely related. The roast that tastes boldest has the least of the compound doing the most biological work. This is, honestly, a fitting summary for a lot of what I cover here. The version of the thing that gets marketed as most powerful is frequently the version that has had the most useful parts removed. Your cells are running the maintenance program every night. Your cortisol is trying to prime your metabolism every morning. Your adenosine is trying to give your caffeine something to work with. Your gut is trying to buffer your blood glucose with polyphenols you could be delivering in the right conditions. None of these systems are broken. They are waiting for the timing they need. The 3-window reset does not add anything to your body. It removes the interference that has been preventing these systems from completing what they already know how to do. If you are going to start the 3-window reset this week, comment window below so I can see who is actually doing this. And if you have not subscribed yet, this channel is exactly this. The biology that is running whether you understand it or not, and what becomes different when you do.